So in this section, we're going to talk about fermentation. This is an anaerobic harvesting of energy. At aerobic, if you have aerobic exercise, that means that you're exercising and breathing hard. You're breathing lots of oxygen. Anaerobic means without. So this is without oxygen. And before I asked in the last video, what happens to the electron transport chain without oxygen? Well, it doesn't function. So the cells then are left with glycolysis. They can't go into the Krebs cycle and into the electron transport chain and oxidative phosphorylation. So the whole point of this is that it does not require oxygen. It's a lot less efficient. We're going to see a lot less ATP made. And it's used by muscle cells, yeast, and some bacteria. And it allows them to make ATP by glycolysis. So we're only going to make 2 ATPs, right? Not the abundance of energy we were making before. So as you'll recall, what happens in glycolysis is we have a 6-carbon chain, and it becomes 2 3-carbon pyruvarates. We make 2 ATP, and we also make 2 NADHs. But now the NADH doesn't have any place to go. Before, the NADH went into the electron transport chain. It could drop off its electrons there, but it can't do that anymore. And for this reaction to occur, you have to have the NAD+. And so fermentation is mainly just a way to regenerate the low energy form of NADH. And there are 2 different ways to do this. The first one here is going to regenerate the NAD+ with this reaction here and make 2 lactate. This is what your muscles in your cells in your muscles do when you're running hard and you can't get enough oxygen to your cells. They will go into just doing glycolysis until you can breathe heavy and get enough oxygen into your tissues. And the other method is to take the pyruvate and make ethanol. Both of these processes regenerate the NAD that you need for glycolysis to proceed. Okay? This is the reason that when we make beer or wine, we have to close them up in a sealed container because we're limiting the amount of oxygen that's getting to them. Otherwise, they will not do this process and make ethanol. So wines have to be sealed, and beer kegs are also sealed for that reason. If you expose the fermenting yeast to oxygen, it'll slow and stop the process, and it'll switch back to aerobic respiration. When the fermentation is rapid, carbon dioxide produced drives away the oxygen immediately above the wine. However, as fermentation slows down, the wine must be sealed to prevent oxygen exposure and permit the fermentation process to finish. So there are 2 types of organisms that use this. One is facultative. Facultative anaerobe uses oxygen if it's present. If it's not present, it's able to use one of the 2 processes above to make lactate or ethanol. There are also organisms that are obligate. That means they are obligated to not have oxygen. They require anaerobic conditions, and they're poisoned, actually, by oxygen. So let's look at the connections between metabolic pathways. You obtain most of your calories as carbohydrates, such as sucrose, disaccharides, starch, polysaccharides, and fats, and protein. Your body has pathways to attain all these different carbohydrates, such as sucrose and fructose and starch and other polysaccharides, and make them into simple sugars. The simple sugars can then feed into glycolysis. This was actually supposed to be carbohydrates. And then sugars goes down below, just to make it. So carbohydrates can be broken into sugars. The fats, and then they go into glycolysis. The fats, remember, fat is made from a glycerol molecule. Glycerol can feed into an intermediate step of glycolysis, and the other part was in fats were 3 long fatty acids. And the fatty acid can be broken into 2 carbon fragments that feed into pyruvate oxidation. Proteins are a bit more diverse, so it depends on which amino acid you have, but proteins can be broken down into amino acids, and then the amino acids can join in part of the end part of glycolysis. They can join at the acetyl-CoA step, or they can enter the citric acid cycle. They can all be broken down and made into ATP. It's also worth noting, and let me get a different color here, we'll do blue maybe, that if we want to make sugars, we can do that from glycolysis. The intermediates in glycolysis can be made to make sugars. We can also make fats by taking an intermediate from glycolysis and fragments of acetyl-CoA, and we can make fats. And we can also make proteins in kind of the same fashion. So our food can be broken down into these metabolites, and then we can use the food, the metabolites we've made, to rebuild the molecules that we need for life to continue. Okay, so that ends this chapter. Well done.